Can you hear? Good morning. Good morning. Good morning. Good morning. All good? Hello. Hi. Okay, so everybody has joined. ಓಕೆ ಇವತ್ತು ಝೂಗೆ ಹೋಗಿ ಬಂದಿದ್ದೆ ಅದಕ್ಕೆ ಇದನ್ನು ಹಾಕಿದ್ದಾರೆ ಡೈಲಿ ಐ ಆಮ್ ಅಟ್ ಡಿಫ್ರೆಂಟ್ ಪ್ಲೇಸಸ್ ಯುನೋ ರಾಯ್ಬಾಗ್ ಕ್ರೊಕೊಡೈಲ್ ಅದೇ ಪ್ಲೀಸ್ ಇಗ್ನೋರ್ ಓಕೆ ಪಾಪ ದ ಒನ್ ಹೂ ಇಸ್ ಮೇಕಿಂಗ್ ಕಂಟೆಂಟ್ ಇಸ್ ನಾಟ್ ಫ್ರಮ್ ಬಯಾಲಜಿ ಸೊ ಹಿ ಥಿಂಕ್ಸ್ ಮೇ ಬಿ ಯು ಡೋಂಟ್ ನೋ ಕ್ರೊಕೊಡೈಲ್ ಓಕೆ ಕ್ಲಾಸ್ 2 ರಾಯ್ಬಾಗ್ ಹ್ಯಾವ್ ಯು ಜಾಯಿಂಡ್ ಅಷ್ಟು ಹಿಂದೆ ಹೋಗ್ತಿರಲ್ಲ ಯಾಕೆ ಕಮ್ ಇನ್ ಫ್ರಂಟ್ನ ಯು ಡೋಂಟ್ ಲೈಕ್ ಮೀ ನೋ ದಟ್ ಈಸ್ ವೈ ಯು ಗೋ ಬಿಹೈಂಡ್ ಅಲ್ವಾ ಇಲ್ಲ ಮ್ಯಾಮ್ ಹಿಂದೆ ಕೂತ್ರೆ ನೀವು ಚಂದ ಕಾಣ್ತೀರಿ ಅದಕ್ಕೆ ಹಿಂದೆ ಕೊಡ್ತೀನಿ ನಾನು ಕರೆಕ್ಟ್ ಎಸ್ ಓಕೆ ಫೈನ್ ಸೊ ಲೆಟ್ ಅಸ್ ಸ್ಟಾರ್ಟ್ ವಿತ್ ಸೆಲ್ ಸೈಕಲ್ ಓ ದಿಸ್ ಇಸ್ ನ್ಯೂ ಇದು ಏನು ಹೆಂಗಂದ್ರೆ ಬಟ್ ಟೆಲ್ ಮಿ ಲೈಕ್ ಐ ಯು ರಿಕ್ವೆಸ್ಟಿಂಗ್ ಸಮಥಿಂಗ್ ಐ ಡೋಂಟ್ ನೋ ಪ್ಲೀಸ್ ಟೆಲ್ ಮಿ ವಾಟ್ ಡಸ್ ದಿಸ್ ಮೀನ್ ಎನಿ ಒನ್ ಏನು ಸ್ವಲ್ಪ ಏನಾದ್ರು ಕೊಡು ಅಂತ ಕೇಳ್ತೀರಾ ಅಥವಾ ಐ ಮೀನ್ ಐ ಆಮ್ ನಾಟ್ ಅ ಜೆನ್ಸಿ ಸೊ ಯು ಶುಡ್ ಟೆಲ್ ಮಿ ಸುಂದರ್ ಸರ್ ಏನು ಹಿಂಗಂದ್ರೆ ಯಾರಾದರೂ ಹೇಳ್ರೋ ಐ ಆಲ್ಸೋ ಡೋಂಟ್ ನೋ ಸಂಬಡಿ ಶುಡ್ ಟೀಚ್ ಮೀ ಇಷ್ಟೆಲ್ಲ ನಾ ಕಲಿಸ್ತೀನಿ ನೀವು ಒಂದು ಕಲ್ಸೋಕ್ಕಾಗಲ್ಲ ನನಗೆ ಹಾಂ ಇದರಲ್ಲಿ ಏನು ಕಾಣ್ತಾ ಇದೆ ಅಯ್ಯೋ ಕರ್ಮ ಫೈನ್ ಟು ಆಲ್ ಓಕೆ ಸೆಲ್ ಸೈಕಲ್ ಸೆಲ್ ಡಿವಿಷನ್ ಮಾಡಿಸೋಣ ನೀವೆಲ್ಲರೂ ಜಾಣಾಗಿ ನನಗೆ ಆನ್ಸರ್ ಕೊಟ್ಟರೆ ಐ ವಿಲ್ ಶೋ ಯು ಅ ವಿಡಿಯೋ ಟುಡೇ ಇಫ್ ಎವ್ರಿಬಡಿ ಈಸ್ ಗುಡ್ ಇನಫ್ ನಿಪ್ಪಾಣಿ ಪೀಪಲ್ ಬೋತ್ ದ ಕ್ಲಾಸ್ ಜಾಯಿಂಡ್ ಏ ಸತೀಶ್ ಅವ್ರು ಅಲ್ಲಾಕೆ ಹೋಗಿರಿ ವಾಟ್ ವೈ ಯು ಆರ್ ದೇರ್ ಅದನ್ನು ಹೂ ಇಸ್ ಇನ್ ಅಲ್ಲಿ ರಾಯ್ಬಾಗಲ್ಲಿ ಯಾರಿ ನೀವು ಪಿ ಯು ರಾಯ್ಬಾಗ್ ನಿಂಗೆ ಒಳ್ಳೆ ಎಲ್ಲ ಗೊತ್ತದ ಸುದ್ದಿ ಸರಿ ಓಕೆ ಕಮಿಂಗ್ ಬ್ಯಾಕ್ ಟು ಕ್ರೊಕೊಡೈಲ್ ಬೇಡ ಹಾಂ ಸೊ ಓಕೆ ಐ ಆಮ್ ಗೋಯಿಂಗ್ ಟು ಸ್ಟಾರ್ಟ್ ಸೆಲ್ ಸೈಕಲ್ ಆ್ಯಂಡ್ ಸೆಲ್ ಡಿವಿಷನ್ ಫೈನ್ okay so cell cycle or cell division how do you define the basic sequence of events the basic this is not there Joseph is not there Athani and Joseph illa okay okay sorry Chikodi class 20 and 20 alli yaru Shashank sir it's a merged class ಕ್ಲಾಸ್ 20 ಚಿಕ್ಕೋಡಿ ಇಸ್ ಮರ್ಜ್ಡ್ ಇಷ್ಟೆಲ್ಲ ಜನ ಇಲ್ಲ ನೀವು ಓಕೆ ಸರಿ ಫೈನ್ ಸೊ ಹೌ ಡು ಯು ಡಿಫೈನ್ ಸೆಲ್ ಸೈಕಲ್ ಈಸ್ ಅ ಸೀಕ್ವೆನ್ಸ್ ಆಫ್ ಇವೆಂಟ್ಸ್ ಇನ್ ವಿಚ್ ವಾಟ್ cell will do cell will grow cell will synthesize the DNA it will synthesize the DNA, proteins and many other things and organelles also and eventually it will divide. One cell will divide into two cells. So this sequence of events when cell is preparing itself for division and growing itself and then finally dividing into two parts is defined as cell cycle. And this sequence of events should be coordinated ಅಂದರೆ ಒಂದಾದ್ಮೇಲೆ ಒಂದಿರಬೇಕು ಫಸ್ಟ್ ಡಿವೈಡ್ ಮಾಡಿ ಆಮೇಲೆ ಗ್ರೋ ಮಾಡ್ತೀರಿ ಅಂದರೆ ಆಗಲ್ಲ ಯು ಹ್ಯಾವ್ ಟು ಫಸ್ಟ್ ಗ್ರೋ ಆ್ಯಂಡ್ ದೆನ್ ಡಿವೈಡ್ ಸೊ ದೀಸ್ ಸೀಕ್ವೆನ್ಸ್ ಆರ್ ಕೋಆರ್ಡಿನೇಟೆಡ್ ಆ್ಯಂಡ್ ಹೂ ಕಂಟ್ರೋಲ್ಸ್ ಇಟ್ ದ cell itself will control this sequence of events. Now these events will see one by one how they are broadly classified. Anyone? Cell cycle is broadly classified as two phases. Come on, this is a very easy chapter. Fast. Okay. Hmm. Interphase is again further divided as? This is S. This Allah. This can be written as yes. ಶಾಣೆ ಇದ್ದೀರಲ್ಲೋ. Hmm. This is divided as yes, PMAT. Okay, and this is divided as ಏನು ಇಲ್ಲ ಬಟ್ ಯು ಕ್ಯಾನ್ ಸೇ ಸೈಟೋಕೈನಸಿಸ್ ಇನ್ ಆನಿಮಲ್ಸ್ ಆ್ಯಂಡ್ ಸೈಟೋಕೈನಸಿಸ್ ಇನ್ ಪ್ಲಾಂಟ್ಸ್ ಸ್ವಲ್ಪ ಡಿಫರ್ ಆಗುತ್ತೆ. ಯು ಕ್ಯಾನ್ ಸೇ ದ್ಯಾಟ್ ಕರೆಕ್ಟ್. ವಿಲ್ ಸಿ ದ್ಯಾಟ್ ಆಮೇಲೆ ಅಲ್ಲಿ ಬಂದಾಗ ಹೇಳ್ಬೋದು. ಓಕೆ, ಲೆಟ್ಸ್ ಗೋ ಒನ್ ಬೈ ಒನ್. ಇಂಟರ್ಫೇಸ್ and M-phase are the two major events in which cell will take part. Interphase further divided as 3 more phases. Is it okay, people, if I go a little bit faster? Is it okay? No, Andre, no, it's okay. You should tell me directly. Illa, ma'am, we need to go one by one. It's okay. Okay, fine. So interphase will be seen as a 3 subdivisions, and M-phase will be seen as karyokinesis and cytokinesis. What is the meaning of karyo and kinesis? If I've split this word, what is the meaning? Karyo and kinesis meaning, you know? Huh? Hey, wait. Which class is speaking? Is it Chikodi speaking? Yaar matadligittiri? One of you, tell me.
Genesis is the process of nuclear division that occurs during cell division.
Nuclear division. Okay, good. From now, whoever answers, this is a reward. Okay, yes, nuclear division. Then cytoplasm division, correct? So, which is a, yes, which boy is answering? Hele answer ಮಾಡಿ that you are answering so that I'll get to know somebody is answering ಅಂತ. Then this 4 divisions, prophase, metaphase, anaphase and telophase, there we'll see only the content of nucleus is dividing. Content of nucleus only. And here the actual whole cytoplasm along with organelles, everything will get divided. Okay, so I am starting with our interface today. Visible this diagram? So, you can see G1 phase, S phase and G2 phase. Interphase is also called as preparatory phase, which is one of the longest. The majority of the cell spends its time in interphase only. Why? For example, human cell approximately takes 24 hours. Yeast cell, for example, how much hour? 90 minutes. Then E. coli, bacteria generally 20 minutes. So 24 hours consider ಮಾಡಿದ್ರೆ ಇದ್ರಾಗಿಂದ 95%, which means 23 hours will be spent during the for using interphase only or preparatory phase only. Why? Why you think a cell does this? It's like exam preparation, people. ಈಗ ನೀವು exam preparation ಮಾಡಬೇಕಂದ್ರೆ ಇಡೀ ವರ್ಷ ಮಾಡ್ತಿರಲ್ವಾ, right? You will prepare for your exams for the whole year. So preparation time is always the larger. ಅದು exam ಕೊಡೋದು 3 ತಾಸಿಗೆ ಅಷ್ಟೇ, right? So imagine your exam is like a one division, andre it is M phase, and the whole preparation is like a interphase. So where you have to do lot and lots of work, where cell only grows, cell will only grow in size but does not divide. Grow in size but does not divide. So first phase, G1 phase or gap 1 phase or growth 1 phase. You can say anything, gap 1, growth 1. Okay, so this phase is again majorly requires the proteins, RNA, every raw materials, enzymes that is required for DNA synthesis is produced in the G1 phase. Joseph, you have joined? Every raw material that is required for S phase is going to be get produced here. My quick question to you, yesterday we saw about histone, non-histone proteins, remember? What do you think histone proteins L form agbaku? Don't you think it should form here only?
Yes.
Because mund DNA divide admele you need histone proteins for packaging, for condensation. So I think they should be madle prepare ಮಾಡಿ ಇಟ್ಕೊಳ್ಬೇಕು ನಾವು, correct? Agree? Then S phase where actually DNA synthesis happens. DNA makes its own copy. If you remember, yesterday we saw that ಒಂದೇ chromatid ನೋಡಿದ್ವಿ ಅಲ್ವಾ? Correct? And after the S phase has completed, you can see one more chromatid, which is an exact replica. Replica means copy, exact copy of the own whole DNA. I hope you remember the difference between DNA, chromatin, chromosome, and gene. Yesterday we discussed. Anybody can tell me DNA, chromosome, chromatin. DNA is deoxyribonucleic acid. Fine. Chromosome, chromatin, at least tell me that. We did this yesterday only, people. Chromosomes are?
In condensed form.
Ulta ಹೇಳ್ದಪ್ಪ. Again ulta. Chromatin is decondensed form of chromosome and chromosome is a condensed form of chromatin. Got it? DNA only when gets condensed forms a chromosome. Chromosome which gets decondensed forms a DNA or chromatin. Clear? Don't forget. Alro PU2 ನಾನು ಕಲಿಸೋದಿಲ್ಲ ನಿಮಗೆ, ಈಗ ಹೇಳ್ತೀನಿ. I will not teach you. ಈ ಥರ ಇದ್ರೆ ಎಷ್ಟು ಬೇಸಿಕ್ ಐ ಆಮ್ ಟೆಲಿಂಗ್ ಯು. ಎಸ್ಟರ್ಡೇ ಹೇಳಿದ ಕ್ಲಾಸ್ ಇವತ್ತು ಮರೆತುಬಿಡ್ತೀರಾ? Please try to remember. If you don't remember, note it down. Barditkolli, no problem. Okay. Then what is the difference between DNA and chromatin? Come on. Try. Chikodi classes, can you try? Chikodi class 20, you are 2 classes merged. You should have a double answers. 4 mic ಇರಬೇಕು ಆ ಕ್ಲಾಸಲ್ಲಿ at least. No? Chikodi class 20, where is the mic? Where is the mic, class 20 girl? ಆಯ್ತು, ಯಾರ ಹತ್ರ ಇದೆ? Answer me. Basic difference between DNA and chromatin. Anyone? No? Don't know andre don't know ಹೇಳ್ಬಿಡಿ. Okay, DNA is deoxyribonucleic acid. When DNA is attached with histone, non-histone proteins, it is only called as chromatin. Adhe DNA ge chromatin anta ಹೇಳ್ತೀವಿ. Adhe chromatin, when it gets condensed, gets a shape of chromosome. Now clear? Remember this. Okay? Fine. So in synthesis phase, what is going to happen? We are just making copy, which means 2C content irodu. It becomes 4C. Yes, the content develops, but the chromosome remains same. The chromosome number remains same. What does this mean now? If there are 23 pairs of chromosome, let's say before replication, idu one pair. Okay? Idu second pair. See, I'm going very basic now, okay? Be very attentive. Ithara, how many pairs the human has? Human cell has how many pairs? Come on, quick answers.
23 pairs.
This is before synthesis phase. Once the synthesis phase is done, every pair will have extra chromatid ethera. The chromatid has become extra. Chromosome number remained same only. Content doubled. Got it? Okay. And lastly, G2 phase. What is there in G2 phase? Again, proteins and organelles also. Some organelles will also be formed here. Andre mitochondria, ER, they will start also dividing. Even this will also start dividing. And whatever protein and enzymes are required for the M phase, that is correct. M phase in Beko, that will be formed in growth 2 or gap 2 phase. So you can see G1 phase, S phase and gap 2 phase whole consists of interphase, and then you have the M phase. This part is clear. Check your books, see if anything not clear. Joseph, what is she writing? First bench. Okay, good to go. ಒಂದೆರಡು ಕ್ವಶನ್ ಮಾಡಿಸ್ತೀನಿ, ವೇಟ್, ದೆನ್ ಐ ಆಮ್ ಫೇಸಿಂಗ್ ಹೋಗೋಣ. Easy questions. This chapter has very easy question. 2 to 3 questions you can expect from this chapter. 3 antu average baratte. Wait. Let me ask one by one. Chikodi class 19 alla, class 19 alla. Innondu yavadu class adu? 22. Chikodi class 22. One by one. Others, please raise the cards. Easy question ಇದೆ. Tell very fast. These are very easy questions. Hange ಹೇಳು. There is no mic. Sudharani ma'am, there is no mic. Class 22 mic ilva? Then at least say that, no? Students, illa mic, other ma'am, illa anta. You should tell that. Adunu teacher ಹೇಳ್ಬೇಕಾ? ಹೇಳಿ, fast. Most of the time, D or B, 2 or 4? Okay, yes. Who is answering this? Okay. What did I talk here? When I say here that maximum phase was spent in the interphase, but still you are answering me D phase. Now you only tell me, students, do I do anything wrong here? Did I not tell you clearly? Did I not inform there? Why are you not attentive? You should be attentive. What I told, the maximum phase, the maximum time the phase spends is what? Others? Interphase. 95% of the time of the cell division, let's say 1 hour cell division ittandandre, then 50 minutes it will do only interphase. Last 5 or 10 minutes it will do the right exact M phase. Okay, is it clear, class 22? Now clear? Do not do mistakes, okay? Be little bit careful. This is our very easy questions. Okay, fine. Class 22 again, same. Try to answer. Others, scan the card. Joseph, your answers. Class 22 again, you only. I'm asking you only. Give this answer. This is a reverse case. Option? M phase. ಉಳಿದಿದ್ದು ಏನಿರುತ್ತೆ? M phase, when majority of them is spent in the interphase, then the rest of the time would be spent by the M phase. Easy questions. Okay, again, very easy. Nipani class 18, anyone from you? Class 18 Nipani, anyone from your batch? Mike Elada? There is no need to see the book also, I guess. Alva, don't you think it's so easy?
Option A.
Okay, where is the mic in class 18? Mike ಯಾರ ಹತ್ರ ಇದೆ?
This answer is from Nippon only.
Oh, no, wait. I'm asking 18, class 18. Hold on. They are not ready to speak, ma'am. They are not ready to speak.
Answer ಮಾಡತ್ತಿದ್ರಿ.
ಆಯ್ತು ಬಿಟ್ಬಿಡಿ, forget it. Yes, the other class, who is answering? Class 19, no?
Yes, sir. Yes, ma'am.
ಏನು ಹೇಳಿದ್ರಿ ನೀವು?
Option A.
Yes, very easy. Try this. Raise the cards now. ಯಾರು ಹೇಳೋದು ಬೇಡ, ಎಲ್ಲರೂ ಕಾರ್ಡ್ಸ್ ರೇಸ್ ಮಾಡಿ, ಓಕೆ? Come on, scan your responses fast. A, B, and C. There are confusion in all three options. Joseph, your answer. ಯಾಕೆ ಮಾತಾಡ್ತಾ ಇಲ್ಲ ಇವತ್ತು ಜೋಸೆಫ್? ಇಸ್ ಮೈಕ್ ನಾಟ್ ವರ್ಕಿಂಗ್? If you are speaking, then it's not audible, okay? Class 20, Chikkodi, anyone? Ha. ಯಾರಿದು? ಆನ್ಸರ್ ಬೇಕು. ಗಿವ್ ಮಿ ಆನ್ಸರ್.
Option C.
ವೇರ್ ಆರ್ ಯು? ಎಲ್ಲಿಂದ ಮಾತಾಡ್ತಿದೀಯಾ? Your name please?
Varzia.
Varsia, okay, yes. Option C. If the cell has 14 chromosomes at interphase, then how many chromosomes will the cell have? Varsia, stand up. Anybody wants to give another answer? To Varsia, what happened? Why option C? Why are you reducing?
Yes, option A.
Okay, option A yaar heliddo? That's fine. Who is it?
Joseph again.
Joseph, why are you messing with the answers?
Option B, ma'am.
Chromosome number, yes, chromosome number remains as it is.
It will be double. In G1 it will be remain same. Option B. Option B.
एक मिनट, G1 इरली, G2 इरली, याद बेखाद interphase इरली, chromosome number remains same until and unless it is a meiosis. Chromosome number is not dividing. Please be careful. Listen. See, M phase we will again study as mitosis. Now pay attention, mitosis and meiosis, okay? So be very clear in your mind. Mitosis is a equational division. Here also chromosome number still remains same. If you have 14 chromosome, daughter cell will also have 14 chromosome. But if we are talking about meiosis, and if we are talking about meiosis and the diploid cell has 14 chromosome, then the daughter cell will have 7 chromosome. So, ಈ question ಅಲ್ಲಿ it doesn't matter whether it's a G1, G2, irrespective. Irrespective if it is mitosis and interphase, still the chromosome number is going to be the same only. Getting any more clarification? If you want, tell me right now. It's okay. Do mistakes by scoring. Yenagala. Okay. Now, if there are 30 chromosomes in interphase, then how many chromosomes are left at prophase? Listen. If I say there are 30 chromosomes in the interphase, then how many chromosomes are there in a prophase? 30 only. Ignore none question only. I have not mentioned whether it's a mitosis or meiosis. Yen bekkadirli. Prophase is the first phase, starting phase. Alli same number ulibeku. Unless there is a cytokinesis, chromosome number divide agalla. Getting what they're writing, Anusha ma'am, what is they are writing? What Joseph people is writing so much? Any assignments? I did not give any homework. I'm a good teacher. ಮತ್ತೇನು ಬರ್ಲಿಗತ್ತೀರಿ? What they're doing, ma'am? ಹೇಳ್ರಿ.
Some of them are writing the questions.
No, you get the PDF only, na? Why you have to write? You get the whole material.
Yes, miss.
Katti kelsa, don't do donkey work. Be smart. Don't do repetition. Something which you don't know, and if I've written on the board, then write. Question, your wish. If you still want to, I'll not stop you. But bad anta ಹೇಳ್ತೀನಿ. ಕೇಳಿದ್ರೆ this. ಕೇಳಿದ್ರೆ okay. And now don't make mistakes, huh? Scan, don't say the answer, just scan your responses. Raibag will give me the answer now. ಕಾಣ್ತಾ ಇಲ್ಲ? ಯಾರಿದು? Is it Abhishek? Who is answering me? Which boy is answering? Nin ಹೆಸರು ಕರಿತೇನೋ? Adarsh? No. Omkar, where are you? Which Raibag? Is it Raibag, Omkar? Huh. Okay. What's your answer? You said? Hello. Answer ಏನು ಹೇಳಿದೆ ನೀನು? 2 or 4? Tell like that. Because B and D ಗೊತ್ತಾಗಲ್ಲ. 4. Huh. Another girl, some another. Hey. Cheating, huh?
Ma'am, second one.
Wait. Which of the following represents the DNA content and chromosome number respectively after S phase in a diploid cell if initial amount of DNA is 2C? DNA content first barbeku and then chromosome number. You are telling?
Second one.
Yes. DNA content initially in a diploid cell was 2C and then it becomes double and diploid cell remains diploid only. Yes, agreed?
Yes, miss.
But there are some people who has given me option 3 also. Yake? Why? Any doubts here? No, na? Can I go ahead? ಹಂಗೆ ಹೇಳ್ಬಿಡಿ. Tell me like that.
Option B. Second option.
ಯಾರು ಅದು ಮಿಸ್ ಅಂತ?
Option.
See, which of the following events occur during G1 phase? Replication, growth and normal function, centriole duplication. D? I will go with D.
Miss.
This is, idu ide. Even growth will be also a part, but More appropriate is DNA and centriole duplication. Where does the centriole duplicate? Which phase? No, S phase. Eh. Centriole, ನಿನ್ನೆ ಹೇಳಿದೆ centriole ಏನು cytoskeletons, two centrioles form the one centrosome. Centrioles are required to form spindle fibers. Spindle fibers are the protein structure that holds the chromosome together and pulls them away towards the poles. Remember? Okay, before I go to mitosis now, I have not told you this, but try. ಹೇಳ್ತೀನಿ. ಈ ಚಾಪ್ಟರ್ ಆಗಿಲ್ಲ ನಿಮಗೆ, ದಟ್ಸ್ ವೈ ಐ ಅಸ್ಯೂಮ್ ದಟ್ ಯು ನೋ. ಹಾಂ, ಹೂ ಇಸ್ ದಿಸ್? ವಿಚ್ ಬ್ಯಾಚ್? ಹಾಂ, ಚಿಕ್ಕೋಡಿ 22. ಯು ಆನ್ಸರ್ 4? Don't hesitate. It's okay. If mitotic division is restricted after G1 phase of cell cycle, then the condition is, yes, what is G0 phase? Also called as, one more name? Quiescent phase. So, now I'll tell you one more concept here. Remember, pay attention. Now what is going to happen in the cell cycle? Idan nodidula now, that diagram. Here in the cell cycle, after G1 phase, sometimes the cell exits the division and enters the G0 phase. It exits the cell division process and enters the G0 phase or G0 phase or quiescent phase, anything you can say. The reason behind that is, the reason behind that is, now this cell will only do the normal functions but do not divide. It will not do under division. This type of cells are called as, anyone knows the name? Differentiated cell. Such type of cells are called as differentiated cells. Iga, I'll tell you for example, your skin cells. Okay, this skin cells, once the whole body is formed in the fetus, they were stopped dividing. That means there will be no cell division, but normal functioning of the cell, whatever cell function is to be done, that it will be keep on doing. So when cell is exiting the cell cycle and doing the normal function, we will say it is a differentiated. Now for some reason what has happened, there is a cut in your body. What happened at that time? You will bleed, there will be a wound, there will be a scar. Now due to that cut, the point at which you have got injured, that cells will receive a signal that you have to redivide, which means cell will enter back into the cell cycle. It will enter, athwa I will write re-enter. It will re-enter the cell cycle or cell division. ಎಲ್ಲಿ ಎಂಟರ್ ಆಗಬೇಕು ಅದು? ಅಗೇನ್ G1 ಫೇಸಿಗೆ ಎಂಟರ್ ಆಗಿಬಿಟ್ಟು S phase. It has to go to the S phase now. G1ಗೆ ಎಕ್ಸಿಟ್ ಆಗಿತ್ತು. It will again enter the S phase, divide the DNA, and prophase, metaphase, anaphase, telophase, everything it will do. Until when? Until the whole injury is been covered. And then once the whole injury has been covered, once it is healed, again the cell will exit the cell cycle and becomes or gains the function. So now when the cell had first time exited the cell cycle and became differentiated, means it had gained the function. Next, when it re-enters the cell cycle due to some signal, due to some condition, that time we say the cell is re-differentiated. The cell is re-differentiated. And now, sorry, D. And then Once again it exits the cell cycle and becomes differentiated, then we say it is redifferentiated. This is not there in your book because this concept is applicable when you come to your PU2. So I am telling it now only. Basic ide, what is happening? Innondu example kodtini, very easily you can connect. You know that the tail of a lizard is cut off and it can regrow. Alva nodidira? Have you held that? So what happens? Suppose this is the tail. I'm drawing only tail, okay? Understand, this is a tail only. For some reason, cut aito. Isht cut aito. Now what will happen? Initially, when tail was formed, imagine that tail was formed of a lizard, all those cells divide agi, it had got differentiated. All these cells has got differentiated. For some reason, tail chopped off. Now, the cells from here got a signal that you have to again divide. So, redifferentiated, they will again start dividing until it reaches the original length. Once the whole original length is reached, again they will stop the cell division and gets redifferentiated. This is what I'm trying to explain you. Got it? So, depending upon the signal, few cells can go back to cell division, few cells. But there are some exceptions who once divided cannot redivide or dedifferentiate. Can anyone tell me those are which cells who in a lifetime, if lost, cannot be redivided? You know that, think. Nerve cell, then cardiac cells. These cells, once if you lost it, if you get damaged it for some reason, fully formed cell tirga divide agalla. Nervous system, if it is losing any of its neuron, it will not divide again. So these are some exceptions which will not divide. Most of the other cells can divide, they can heal at certain level, but beyond that, other cells like cardiac cells and nerve cells will not. Okay. ಇದು ಒಂದೆರಡು ಕ್ವಶನ್ ಇದೆ, ನೀವೇ ಮಾಡ್ಕೊಳ್ಳಿ. Just now I told you this. Come on. Which of them cells in adult animals do not exhibit division? Exhibit division andre ಏನು? First time ಏನು form ಆಯಿತು heart when it was a baby, when it was a fetus. That's it. Now in the whole lifespan it cannot redivide again. Not possible. Getting? I don't think so. You will be able to hear people. Allah, you can't hear, na?
No, miss. Yes, ma'am. Can't hear.
It's okay. It's okay, you will not able to hear this because e-speaker all connected illa. This is the mic. So it is not connected here. You will not be able to hear it. Leave it. It's okay. This, this video is about mitosis. Because you have already done that, let me just show you the first video and then we will go through the phases. Athwa first phases madli, how it is okay for you? Okay. Mixed answers. Fine. Observe. Silently observe this, then we will go through it. You will not listen it, but you can read. Innangu one interesting question, ro. Wait. Let me see who can answer this, okay? Why it is called daughter cells always and why not son cells?
Women are capable of dividing.
Arey wah. Very good. That's correct. Who is this? Whoever is this?
MyPAL-Phygital.
Thank you. Great answer. Okay, go ahead. So lonely. How do you grow from one cell to the whole human being? How the tissue repair is happening, they are seeing. See, adane ಹೇಳಿದ್ದು ನಾ ಕಥೆ ಈಗ. Differentiated, dedifferentiation, redifferentiation. What is that dark dot? Nucleolus. See, these are what? Chromatin fibers, the DNA. Yes, it will get condensed now. DNA is getting condensed. ನಿಮಗೆ ಇನ್ನೊಂದು ಮಾತು ಹೇಳ್ತೀನಿ ಪೀಪಲ್, ಇದು ಹಿಂಗೆ ಸೆಪ್ರೇಟ್ ತೋರಿಸ್ತಾರಲ್ಲ, in actual DNA is a one long strand, okay? You should never think that DNA becomes small pieces like noodles. No, ಹಂಗಾಗಲ್ಲ. ಅದು ಏನಾಗತ್ತೆ, at some places it is tightly coiled, so it looks like this. ಇಲ್ಲಿ connection ಕಾಣಲ್ಲ because it is very thin, lightly stayed, that will not be able to visible to our